All right, so we'll begin. So welcome everyone to Straight Science. This is live Straight Science. It's a hybrid. You're on if you're online, and we're here in the room here. And we're a little rusty post-COVID with a new system with the here at UAF Northwest Campus. So welcome everyone to Straight Science. Straight Science is a public evening science presentation series put on by UAF Northwest Campus, this room here, and also UAF Alaska Sea Grant. Both UAF Northwest Campus and UAF Alaska Sea Grant, we are public servants of the Bering Strait region. And the Bering Strait region is the homeland and waters of the Yupik, Inupiaq, and St. Lawrence Island Yupik peoples. So tonight, you may have, in a blink of an eye, you may have seen Sikuliaq come into Nome yesterday, and they turned right around. So they were doing a crew change, I believe, and they were doing a research change. So it was pandemonium at the West Jetty yesterday afternoon. There was some delays getting them in even, so they were really short on time. They did a great job switching everything around, and tonight they're on their way. They're just sort of north of Nunavak. And if they're listening, I don't know if they're tuning in for this, but people here say hello to Sikuliaq, of course. And we have two speakers. Neither one is a stranger to Straight Science, and we're excited they're here. They just got dropped off last night, and they're making a point of letting us hear in the region what they saw, what's going on, why they were out there. So I will stop talking here for a minute. We're going to have Jackie Grebmeier with the University of Maryland's Center for Environmental Science. Again, no stranger to the Bering Strait region. She has been, what's the polite way to do it? You've been aged. She's been doing research in the Bering Strait region for decades. Yes, for decades, looking at the seafloor sediment and the animals in there, and all the ecology that goes with that. And we also have, very excited, Seth Danielson from the College of Fisheries and Ocean Sciences, University of Alaska Fairbanks from Anchorage. And they always seem to save a seat for him on Sikuliaq. So it's very nice that he is here to also let us know what his work was, what they were seeing, and so forth. With that, for the audience, I think I'm supposed to let you know that there are two exits in case of an emergency. You can get out of this room and go right or left to the doors. And because we are live, if you need to use the bathrooms, they're just around the corner. Just take it right out the right and go, you'll find the signage. With that, welcome and take it away.
Oh, thank you, Kate, so much. And it's all good to be back here in Nome. Thank you for helping facilitate that, and for the people in the room and those who are online. Seth Danielson will be talking. We're doing a joint talk. We were co-chief scientists on the Sikuliaq. So as he said, I'm a biological oceanographer from the Chesapeake Biological Lab at the University of Maryland.
And I'm a physical oceanographer. Dave tried to assign me to Anchorage, but actually I'm from Fairbanks.
So did I say? I'm so sorry. Yeah, Fairbanks.
I figured it was on the slide, so you'd let her know.
Yeah.
So we're going to be team doing this, and so that we'll be doing parts of it. I'll do the introduction, and then Seth will come in, and I'll do biology, so we'll do the physics part. Okay, so we're off.
Take it away.
All right. So thank you all. Yes, we're now at the Sikuliaq. We started in Dutch Harbor in the 10th of September, and we got off here Nome yesterday. And you can see the Sikuliaq in the upper left part, and we're looking. We did a joint program with 4 other. If I can advance this. We had this all in practice. Where is it? Oh,
We need the mouse.
the mouse.
Try that.
Oh, there we go. Upper left part. So it's a consortium basically of 4 programs. We have the I've talked previously about the Distributed Biological Observatory, where we've been doing anywhere from 10 to 30 years on these time series from south of St. Lawrence Island up to Tiavik. And this is a combination also with the Ecosystem and Fisheries program of NOAA called EcoFoci, and then we're collaborative with the Arctic Marine Biodiversity Observing Network called AMBON, and the Chukchi Ecosystem Observatory in the Northeast Chukchi Sea. So while we're doing this, we're looking at ecosystem status and trends, and some key questions: what's happening with the system? How are the changes occurring? What are the key drivers for that? Important things that we're also looking at are everything from the food web, from the microbes to the whales. We're looking at food security topics, so we're looking at harmful algal blooms, the impacts throughout that food web, and also ocean acidification, because that's having an impact, a lot of CO2 in the bottom waters, and that has an impact on certain organisms. Well, now it works. I just need to be woken up, I think. So there's a lot of information here, but main thing I think that, as I've already said, what we're looking at is status and change, standard measurements. We go from physics, we do chemical measurements, looking at nutrients, oxygen, a lot of parameters that you see up there, but we'll be talking about each one of them in the presentation. We look at water column phytoplankton, zooplankton, larval fish. We go down to the benthos to look at the animals that live on or inside the sediments, and they're very important then for big animals, marine mammals, seabirds, fish that are feeding on them. And then we have seabird and marine mammals from the bridge. An important point to see over here on the right, and I don't see an arrow, but there it goes. And so we started off in Dutch Harbor here. We went up. The DBO stands for the observing system. We have time series sites southwest of St. Lawrence Island, and embedded in these are moorings, and Seth will talk about that, where we have time series measurements with a lot of sensors, sediment traps, marine mammal acoustics on them. We went up then into the south to the Chiricahua Basin here, did set sampling there. And then we shot all the way through Bering Strait to be up by and working off the northeast Chukchi Sea at Utiavik before the hunt began. Subsistence hunt, we wanted to make sure we were away from the coast. And so we succeeded in doing all of this effort, moving down and then coming down and working south, and then ended up doing DBO3, and then came through and did a line across Bering Strait, marine mammal mooring at this NM1, and came in yesterday. So with this, I'm gonna let you talk about nitrate.
All right. So I've got a map here with some arrows on it, which show sort of the average flow directions that of the major current systems across the Bering Sea and Chukchi Sea continental shelves. And one of the interesting things about this region is that the Pacific Ocean stands about 3 feet taller than the North Atlantic Ocean, and water wanting to flow downhill initiates that huge flow northward through Bering Strait, about 1 million cubic meters per second flowing north through Bering Strait on average, and that's helping close the world's freshwater budget. Well, that nutrient-rich flow of the NIGR current, which is shown in the orange.
You admit.
Oh yeah.
Yeah, I don't know why it's showing up on yours.
Yeah, there we go.
Which worries me, but.
This orange arrow here is a pathway for incredibly nutrient-rich waters from the continental shelf break of the Bering Sea into the inner shelf, the northern Bering Sea shelf, through Bering Strait and into the Arctic. And fundamentally, this is why the Bering Strait region is so biologically productive. There's a massive input of nutrients that occurs year-round. So it's not just during the time of spring bloom when most systems get their big pulse of nutrients through the year. The Bering Strait region actually gets nutrients continuously, and that allows for massive phytoplankton productivity, zooplankton, and upwards through the food web, so that there are these fantastic populations of seals and whales and seabirds in this area. So as the ship is driving around, this is the ship track that we sailed. We have a number of sensors that are making measurements continuously every few seconds. And this sensor here that we plotted is nitrate. So that's one of the major nutrients for the phytoplankton growth. And you can see that just north of NIGR Strait, there's a lot of energetic mixing that goes on in NIGR Strait. And those nutrients are being brought up to the surface. The ship makes its measurements of these about 5 meters below the surface. Massive amounts of nutrients right there. Even here at the end of summer, typically when there wouldn't be in the waters. And so it's sort of eternal spring bloom up here. We can always have phytoplankton productivity if there's enough sunlight coming in.
You want us to admit, Kate?
Oh,
Yeah, I don't
there's
know what-
more people coming in, sorry.
Well, they're in already, and so I'm unsure of why we're getting this, because I am not shown that option, so.
As long as
Okay,
they're
keep going.
legal.
Continue on, we'll
Okay.
figure it out.
Sounds good.
Thank you very much.
Okay, so nutrients are flowing in and we're also measuring things like temperature and salinity as we're driving around. So this is the habitat in which our marine ecosystem is living. So very fresh waters near Nome and the outflow from the Yukon River. Cooler waters associated with that mixed up water from the Onondaga Current. Cool waters in the northeast Chukchi Sea. Here's the sunlight, so photosynthetic available radiation. This is the amount of sunlight that's coming in, available for phytoplankton to turn that into biomass. And on the right-hand side here, we see localized hotspots of chlorophyll A. So these are concentrations of phytoplankton that we're driving around and finding. So this is some of the sort of environmental setting data that we're collecting as we go through the cruise.
Photosynthesis, that was all blue, which is 0.
So
This is September.
this is September. It's not 0, but very low levels in the near surface. But when we get down below the surface, you'll see in a couple minutes that there's actually some higher levels down deeper. And one bit of chlorophyll that we saw was associated with a bloom of coccolithophorids in the southern Bering Sea. As we steam north from Dutch Harbor, coccolithophorids have this characteristic milky white color in the water, and it shows up the same way in the satellite. This is a true color satellite image, kind of difficult to see, but here's the Alaska Panhandle, Bristol Bay, Nunivak Island is here. Here's Norton Sound, and there's this wide swath, and this bloom of coccolithophorids has been out in the Bering Sea for a couple months, and we drove right through it and took some water samples there as well.
Could you see that when you drove through?
Oh,
it?
Yeah, I took that picture one morning when I woke up and I said, "Holy cow, we're in
That's
a
it on the
bloom."
left, yeah.
Yeah, that's from the cruise. Okay, so now getting into the work of this cruise. One of the major objectives of the cruise was to recover and then redeploy a number of moorings. Most of them were operated by NOAA. There's a map here showing, I think, all of the locations, just about all the locations where these moorings are. These are oceanographic moorings measuring ocean currents, temperature, salinity, nitrate. There are water samplers on some of these that collect water samples that can then be analyzed for nutrients and DNA. And there are additional moorings that are associated with the oceanographic moorings that are outfitted with passive acoustic recorders. So for underwater sound, particularly marine mammal vocalizations. Anything else we should say about this here, Jackie?
No, I think that the thing I would say, and it's 2 more is in the audience here, but the photo on the right, so they're listening throughout the year on the left. That's where those moorings are. And unless Sue wants to come up here, you can correct me, but on the right whales are extremely endangered species at very low levels. They're protected in that southeast above the Aleutians there, but you're seeing that they're areas that they're even further north. And the concern is in these sonobuoys on the lower right there, where it looks like they're putting a torpedo in the water, so that you can listen for nearby about those organisms. And the concern is for shipping issues, that these animals may be very susceptible to, along with all types of marine mammals, for the increased shipping that goes through this area. So I just put that as one example of what is being done long-term on the time series, short-term when we're out at sea. One other thing I would just like to say on what he had on the environmental, all those original figures of temperature, salinity, those were sensors flow through the system on the ship. We didn't stop at any of those stations. So we used the ship as our sampler. So that allowed us to transit up to the area where we start doing vertical profiles. So it's a really very versatile and a highly technology available ship for that.
So can I ask a right whale question, since we have a right whale person in the audience? those two dots that are right by St. Lawrence Island, do you know if those data points were from the Ashuru Maru cruise a few years back where they biopsied a
I don't
right whale?
think so, Gabe, but I'm not certain. They did have long-term recorders in those
Oh, this is
areas.
recorders.
This is acoustics.
Okay, we often see the, okay, thank you very much.
Does sonar affect whales?
At the right frequencies, Kim, I think you should speak to that. Pam.
I think we can talk about that maybe later. That's a big.
Maybe in the questions,
Yeah.
we'll ask at the question end, might get in depth on that.
Yeah.
Yeah, good question.
So some of the oceanographic moorings look something like this. They've got a variety of sensors on board. Being deployed here as a sediment trap. And at the end of the year, 24 bottles have each been opened for on average about 2 weeks each. And these cups are capturing the carbon, the biological material that's settling down to the sea floor. So it's dead and dying phytoplankton, it's fecal pellets from Zop Plankton. It's the food that's showing up on the seafloor for the clams. And by capturing these, then the analyses in the lab at the end of the year can talk something about the food quality that's showing up on the seafloor, the species of phytoplankton that's showing up. We can get an idea of the timing of when the sea ice is melting and dropping ice algae into the water column. So we've had a number of these deployments for the last 10 years, and they've returned some really rich data sets to us. Another nice thing about the moorings is that they're out there year round. So the ship is only out here for a couple of weeks at a time as the stations are sampled. These moorings are at a single spot, but with instrumentation on board, they're measuring nearly continuously. The temperature and salinity data loggers measure about every 15 minutes. And so here's a 9-year record, I guess, 8 or 9 year record of temperature near the sea floor. This is in the northeast Chukchi Sea, near Hanishal, between Hanishal and Barrow Canyon. And it gives us a means to really understand what the changes are from year to year in a better sense. You can see in this case here, 2021 wound up being a fairly cool year. There was a lot of ice and not a lot of ice melt back during that summer, whereas 2017 and 2019 were notoriously warm. Well, the critters on the seafloor are feeling those temperature changes. Metabolic rates are linked directly to the temperature that the animals are living in. So the amount of food that they need to consume to just fulfill their basic metabolic needs changes with the temperature. And so this is one way of keeping track of some of that. The bread and butter instrument for sampling from the oceanographic ship is called a CTD, and it has that's for conductivity, temperature, and depth, and it has a number of bottles attached to it. And we stop the CTD instrument at different depths through the water column, collect seawater from those depths for analyses in the lab and on board. So I'm going to show a series of plots here that give an idea of what the water column was like at a couple of our DBO stations here. So here in the northeast Chukchi Sea, the DBO4 region, fairly cold water near the seafloor, about -1.5 degrees C. So this is remnant from the prior year. Somewhat surprisingly, it was fresher offshore than right near the coast. Usually we think of the coast as being the freshest waters with the Alaska coastal current. There was actually a fair amount of phytoplankton here at some of the stations, about 20 or 30 meters depth. And the oxygen trace here is a sort of a fingerprint of the chlorophyll and production that took place a few weeks ago or a month or so ago. So there was a very significant bloom happening here. This is not super surprising. We see this every year when we go up at this time. And so even though there's not so much chlorophyll at some of these stations, you see that there's a lot of oxygen, and so there likely was a pretty massive bloom not that long ago. This is up in the northeast Chukchi. In the southern Chukchi off of Point Hope, again, we see very fresh waters offshore, but we're also seeing the coastal current waters close to shore. There were some wind events that were probably moving some things around. What else is of note? This has a bloom near the surface this time in the phytoplankton. Yeah, Gabe.
So I don't know if I missed it, but so to read your graphs, the blue areas are where Sikuliaq stopped and took a sample.
Yeah, so
And then-
the blue dots, thanks, I should have described that. So the blue dots are the stations at which we're sampling. And then these colorful graphs, the vertical axis shows depth from 0 meters down to about 50 meters, so about 150 feet down. And then the horizontal axis shows the distance along that transect from left to right. So this transect is about 120 kilometers long.
So that's like the coast there.
That's right.
The
the-
bottom's coming up.
The bottom is coming up towards the coast.
Okay.
So we've got temperature in the upper left, salinity in the upper right. Water clarity here, beam attenuation, so that's how much light passes through the water, is in this panel. Chlorophyll fluorescence is here, so this shows the near surface bloom that I'm talking about, and then oxygen down on this plot.
Awesome, thank you.
Yes, thank you.
And the color blue stands for,
It depends on which plot you're looking at. So for temperature, blue is around 0 degrees. For salinity, these are the fresh waters.
Like each one of those is a different.
Yeah.
Yeah.
Screen of that red
but not
box.
knowing the colors, I'm hard to.
Yeah, it's hard to, they're small.
It's hard to know.
Right, and oftentimes, just so you know, on all of these graphs, it's that if it's a red and an orange, it's high, it's higher. And if it's a blue and a purple, it's lower, just the gradients, and that's what the comparison is there, no matter what the units are.
So that's and then finally, southwest of St. Lawrence Island. Perhaps you've heard about the Bering Sea cold pool before. We had a lot of cold water down there along the line at most of the stations on the line, fairly warm waters up above, a fairly weak phytoplankton bloom or concentrations there, although again, some evidence that there had been a bloom there previously with the oxygen.
This was September.
So this is September, yeah, after
okay.
the vast bulk of the productivity is happening
Yeah.
in the region.
All right, thank you.
Yes, I'm gonna turn it over to Jackie here for a bit.
Yeah, and I would just say, one of the things we do within our observing program is we have a cruise with the Sir Wilfrid Laurier Canadian icebreaker that we go out in every year. We've been doing this since '98 in July. So this is when we often pick up high productivity, and other cruises do too, because that's usually when, you know, the light is out, if there's still nutrients, it's gonna bloom. So that, but what we're seeing is later blooming in the fall, actually. So seeing some of these indications is something that's happened in recent years. So you now have all this phytoplankton as food, and so you're looking at now the next level, which is the zooplankton. So these are the small animals that flow with the currents. Copepod, this is an example for the copepod abundance and distribution. They're collected with nets. They have set sizes of mesh. One size is usually for population. They preserve that. The other net is for genetics, so they preserve it and put a different preservative in it and do analysis. But on this particular cruise, they also did microscopy right on the ship, so they did a kind of rough and dirty of zooplankton, so we got some data. And you can see on the right, these are abundance of these copepods, and the abundance by size of the circle gives you how many, and I think they range from 200, I can't even see it, 2,000 to 8,000, and these are in per meter square, per meter cube, so it's a volume. And these are, you can see there was a fair amount still in Southwest St. Lawrence. We don't see that in the early spring, actually, because the copepods are just coming out of their life cycles usually. They go deep in the Bering Sea and then they come up on the shelf. And so, but now you're seeing them at this time of year. Really interesting in the Chiricahua Basin right off of Nome, that you can see the highest levels actually were copepod. They're grazing a lot of that phytoplankton. And that has then an impact on what gets down the sediments to feed the animals that I'm interested in. I'm interested in a lot of animals, but for the ecosystem, and then you can see as we move north. So that's one of the components that was collected.
Do you know what species?
I don't have the species
Okay.
of what they, We're,
They weren't trying to differentiate, it was just large and small differentiation,
Ah.
very quick assessment.
yeah, this is the quick and dirty, but they'll do all that.
Yeah.
So you
Cool,
can,
thank you. That's good to see it though. That's.
but the other exciting thing is the euphosid abundance. These are krill. And these are important for organisms, you know, bowheads are eating them, but they're also important for other organisms such as gray whales. And so the idea is that some of these feeding areas, like particularly as you move north, there aren't a lot of what they normally feed in the sediments that we'll show you a little bit. But they're actually, the hypothesis is that they're more of the euphosid's, and so it was great to be able to catch these that are krill. And they're bigger, you can see their big eyes and they're lipid rich. And so you can see the distribution, and we'll have a lot better idea once they do the IDs, what the species are and weights and that type of thing. So for the benthic work, this is the team that we have. They're all in the room here. Lee Cooper and myself, Sadia, Brian and Nick are all graduate students. So we were very happy to have them. They each have their own project. Brian's looking at the amphipod changes and how much caloric change and impacts on gray whale. Nick is looking at biodiversity and genomics. And then Sadia was assistant, but she does freshwater work, but they were very good out on the deck and in the lab. So just some preliminary results. We have been looking at bivalve populations, particularly southwest of St. Lawrence Island, at least I have, our team for over 3 decades, and have been looking at particularly with the threatened spectacled eiders, and there's a Polinia there, which is an open area in ice-covered seas. We are still continuing to see lower production in the southwest sites, which is our key feeding area for these eiders, but they're still stable in 2 of the 5 time series sites we've been looking at. What we're seeing is a contraction of some of these organisms that are more adapted to the colder waters of the Arctic. And it's something we're seeing as we march up, and I'll show you a few of those in the DBO. When we go to DBO 2, which happens to be right off here in the Chirikov Basin, we're seeing a contraction of these amphipods. They're small shrimp. They're critically important very important over decades for gray whales that migrate from Baja up here to feed in this really rich ground. But now the patch that they're used to is decreased, and that's what we've seen changes in grain size, which is a current related thing. And we're also seeing, you saw were highest in the Chirikov, they graze that carbon. So that how much actually reaches the bottom may be less. That's where the food supply is declining even though there's production there, may be an impact on the quality. We'll know this from some of the work that Brian is looking at, because we're trying to get a conversion factor to go back decades to compare. And then the Macoma calcaria is a white clam. It's fed upon by walruses and bearded seals in the southeast. This is now north of Bering Strait. And this is just visuals because we go back and spend months in the lab, but there are large ones there, which is great to see, except they're adults, and there are very few, if any, small ones. So this is an issue of recruitment. And so if there is no young, you don't have any adults later on. And that is something that we're watching quite closely, because that is an important area for feeding by a lot of benthic feeding animals. And all you're seeing is the casting characters of other animals that we study on the bottom. Just one example of results, because a lot of it goes into buckets and we go back to the lab, and that's what keeps us going in the wintertime. Is this a sediment chlorophyll? So we not only look at chlorophyll in the water column, but we look at whatever remnants come down to the underlying sediments. And so they're a seasonal indication of what the export is. And so we're seeing this reaching, it's a food for the animals living in the sediments. That could be clams, worms, or amphipods. And what we're seeing, and we still see all these hot spots here, higher, you can see here, there's 2 northernmost stations, less so down in here, very hot. We are seeing food down in the region, the Chirikov. So you see a trend, high offshore in the hot spot, still low in the coastal water. And so we're using this, and we'll compare it with other components. And we did some experiments on board too, but those data aren't available yet. Biodiversity is a big part, changes with time and with a season, as well as climate impacts on that. Who's living where, and so we collected, this is Katrin Eikens and her team's work, collected over 107, and the plus is there because she was doing animals that live on the surface of the sediment, and our team was looking at ones in the sediment. We all collected different species to have to have some genomic work done on that through the mitochondrial and building a reference library, so we can figure out what their metabolic compositions are. Species richness was quite strong on some of these areas. It ranged from 17, and the IC stands for Icy Cape. That's kind of midway in the Chukchi Sea, off of Icy Cape out offshore, up to DBO4, which is what Seth talked about, up to 56. So that means the biodiversity was a lot higher up in the northeast Chukchi Sea. We also see that near shore in the Alaska coastal water because there's usually less food supply, there's more grain size, and so animals can find different habitats there. So there's tendency of a slightly higher species in the Chukchi in the Bering Sea. And the list you're seeing on the right are just types of animals. There's cnidarians with their sea anemones, mollusks, so you got your clams and gastropods, your snails, annelids or worms, arthropods, these are your amphipods, the small things that gray whales feed on, a lot of other organisms, tiny different types of crustaceans, decapods, your crabs, isopods, and so forth. Echinoderms, these are your sea stars, the sea urchins, sand dollars, sea cucumbers. And then you can see the list all the way up to your closest relative, which happens to be a tunicate called Acidacea, just so you know. You do have relatives in the invertebrate realm. We do measurements of environmental DNA, and that's also to look at biodiversity. And the reason we say microbes to whales is because these are being measured from the smallest to the largest organisms. And they do collections, they have it not only from the water column, but collecting these moorings, and this is new this year. And so the eDNA, well, for example, the collections like when a whale goes past and it has flakes off its skin, or you have a fish and it flakes off its scales, or any organism leaves some part of it, that's being able to pick up some of that through the collections on these moorings, as well as the water column. So we could start fingerprinting basically all different types of organisms, including commercial fisheries that may be moving north also. So this is an exciting development there. Harmful algal blooms, I think you've all heard about that, the toxicity aspects of it. IFCB stands it. It's a instrument
Imaging
phylocytobot imaging, and we had two of them on board. One of them, some of you may have heard, it was being controlled remotely from Woods Hole Oceanographic Institution, and it was looking at surface waters. And then there was weekly report, and if there was any issues, then it was there's a whole policy that's been put together. It would go to the human health folks here first before the information would go out to all the coastal communities of potential toxicity. The other one was a one by NOAA, and it was looking, vertical collections were being made. And so those data are not yet available on that, but I'm glad to say that we had no record of harmful algal blooms on the course of this cruise. But as you all know, in July of this year, there were some massive blooms in the southern part of particularly in I think the Chukchi Sea.
They were actually the highest numbers, just so you know, so people in the audience would know, is that the bloom occurred about the highest bloom numbers were about 75 miles northwest of Savunga.
Of Savunga?
Yeah.
Okay.
In the Bering Strait.
Okay. Yeah, and one of the things about these organisms is that they bloom, then they settle out and they form a cyst, they overwinter. And so they can be brought up at warmer water, and they like warmer waters, up earlier into the euphotic zone to start blooming again the following year. So it's not like they boom and they go away, they just form cysts in the sediment. So we, on our benthic team, we collected sediments for them to look at those cysts. We collected dominant animals, particularly clams, worms that we figured out. And then on this year, we did suspension feeding tunicates that are then being processed to see what the level of HABs would be in those. And then the other components of the food web were also collected. So then we had seabird observations. So there's this Marty Reedy here, the U.S. Fish and Wildlife Service. And so he was up on the bridge with Sue, who's did marine mammal observations. And just some of the key takeaways, there were many loons in the Chukchi Sea that he highlighted on this. There were many kittiwakes near that coccolithid bloom that Seth spoke about.
He said that was kind of surprising.
Surprising,
Yeah.
yeah. I think these were kind of surprising things. There were aspects of it. There were the common birds you could see. We saw spectacled eiders. We didn't have any issues with spectacled eiders or any of these birds coming. Well, we did have a land bird land on the deck, but then it flew away. But it was this overnight visit of a snowy owl that took everybody's attention, because it sat for 4.5 hours on the front part of the ship, and we were offshore, how far?
60 miles.
We were 60 miles offshore, so it was obviously resting.
Where was it?
It was on the icy Cape line. It was on the that rhino horn in front of the.
Yeah, I mean, a huge, so it was a big hit, although it was that dark, but it was white, so everybody's trying to take pictures. But yeah, so that was something to have that snowy owl there. As far as marine mammal observation, so this is the work that Sue Moore, who's in the audience here, they were healthy feeding gray whales. They were in tight groups just south of Bering Strait. And this is in the Chirikov Basin, the DBO2 region, but in the northern part of it, not the more southern part we've seen them. So there's, like I said, there's a contraction of prey base, and then you could see mud plumes, right, for those feeding. She saw many humpback whales along the shoals in the north side of Seward Peninsula. And there are a few whales in the northern Chukchi Sea. So that, but she did see walruses also. You saw seals, right? That type of thing. So
Any sea lions?
no sea lions.
Thank you.
So just so to conclude, some other samples collected. So primary production, and these are the name in the brackets, and the little stars mean that they were early career students, the graduate students working on projects. So primary production was being done. Haley Sinar was on that. Savannah Sandy was working on ocean acidification to look at the changes, the CO2 level, and that is very interesting for those of us who study water, because we can have below what they call 1 omega, where you could be corrosive to some of the organisms in the water. Southwest of St. Lawrence Island, and often north of DBO4 in the northeast Chukchi Sea. So this data will be very interested. It's all processed offshore. The harmful algal blooms, besides having the IFCVs, as I said, we did all these sediment. There were water column collection and key benthic animals, and that was done by Patrick Chapata, who's a postdoc. And so he would have a star by his name. He has an early career out of Nome. Nitrogen cycling with Stewart, and this is a collaboration with the Brits. So it's a program, they're looking at nitrogen cycle in the Arctic and the Antarctic. So this was the first, the work that they wanted to do with the Pacific Arctic. eDNA, Matt Galaska is at the University of Washington, NOAA. I guess all NOAA now. Genomics of prey, Nick, who's in the field there, in the back here. Silverson is doing that. Caloric studies by Brian Marks on the amphipod, so that's the amphipod gray whale story. And there were nutrients, oxygen, and chlorophyll done by Eric Weisgarber and Caitlin McFarlane, and they're out of NOAA. So, there's that's one thing I have to emphasize, is that we had 4 large programs on this ship to combine it, so we didn't have 4 separate ships going out with different programs, and that's and it really does make a very multidisciplinary and cost-effective way to do a lot of different collections. So we were working. The ship ran for 24 hours a day, 7 days a week. People ran long hours, not all 24, but oftentimes just a few cat naps in between. And a lot of the mooring work was done during the day. So a lot of the biological and transect lines were done at night. But it was a very collaborative and cooperative type of cruise. And I would just close by saying we did have outreach. We went to 3 classes today at the Anvil City Science Academy. And you can see there I am, and then Seth is there with classes ranging from 5th to 8th grade. And Sue was there, and Brian and our students, Sadia and Nick, to answer questions. And they had questions afterwards, and it was kind of exciting to do. So we got feedback from them. And so it's wanted just to conclude by saying thank you to captain and crew. They're, like you said, they're right north of Nunivak. We're down there. Maybe doing a turnaround on a mooring for Phil Stavino and the southeast Bering. The marine technicians, everybody on the crew, they were just great. But I also gave, as always, useful. But Alex Whiting, they managed to bring, there was this critical part that we needed, and it was flown from Seattle to Nome. We missed the ship in Nome. It was flown to Barrow, but the weather, they tried to deliver it from UIC to us, but the weather wouldn't let it. They got a ship to Kotzebue, and they took out, how many miles did they come out?
We drove partway there, but Bobby came out and met the ship. It was fantastic.
Right, and they transferred it and they said, we asked them if they wanted a cup of coffee and they said, not with the winds picking up, and they left. So it was great. And then Lindsay Cameron, the UIC. So there was a lot of team effort to get that part. It was part of this eDNA that went on the final mooring and went into the water. So that was all. And we have all the agencies to thank: NOAA, NSF, AOS, ONR, NPRB, Fish and Wildlife, UAF, State of Alaska, for support for the ship, but also for the personnel on board and for the various science programs. So with that, I think we're open to answer questions.
All right, well, thank you, Jackie and Seth. And before we start writing in our questions, this is a good time if you're in the online audience, to give Jackie and Seth show a little love for their efforts. It's never easy being a public speaker, and they've been at sea for the last
25 days.
25 days without a break, and they came off, and here they are letting us know right off the bat what's going on. So if you want to throw something in the chat, that's great. And anybody here, I guess we'll start with people in the room. Any questions?
The ribbon
The ribbon.
on the right hand side.
Oh, here, this thing. I can see the questions if they're coming.
Yeah, but the dots, but that way you'll see the people.
Okay,
Little dots.
yeah. All right, we're getting some good instruction from the audience. Sit tight. We're gonna be able to see everyone.
You do it from the ribbon on the right hand side there.
Sure. Why don't you, I'm not sure what you're asking, so that's confusing. I you wanna see this?
Yeah.
Oh, there it is.
Okay.
That's right. Sorry. That one.
That,
There you go.
there we go. All right. Very good, thank you. That makes it more, and you can see, I guess. That's right, in case someone clicks on. So with that, any questions right off the bat? Lou?
When I was learning about whales, the gray whales were always bottom feeders. Is that still, you were
They-
talking about the mud plumes. Is that, and then you were talking about how they were in the column feeding, and that,
They feed on both. They still feed on the amphipods when they're there, where they're there. And Jackie's work has shown, especially in the Chirikov Basin, that what was a big area with amphipods has shrunken up. When you get into the southern Chukchi Sea, it seems as though the gray whales may be taking advantage of krill coming in. We have more flow through Bering Strait. We don't, and this crew's included measuring krill, but we think that they are perhaps feeding on both ways, in the pelagic, on the krill, and also on the bottom. And I think that might be happening in the northeast Chukchi Sea as well.
and they're as plentiful as they used to be, or are the gray
Well,
whales disappearing too?
Gray whales have been undergoing a mortality event since 2019, so that we know a good number of them have died. And this happened 20 years ago as well, 1999 and 2000.
So it's a singular,
They go up
it's a,
and down.
the rabbit cycle.
Big rabbits.
Any other questions? I know I've got a couple. What was, because Jackie and Seth, you both have been doing this for years, and you've been up this time of year, and we've been having, you know, our weather, just about everything's in a state of flux here for the last couple of years. What was the most unexpected, first Jackie, then Seth, what was the most, what was the most unexpected thing, or was there anything unexpected in what you saw? Because you have the experience of in this region and
Right.
up the trail before where you went on this ship.
Well, I have to knock on the weather aspect. It was one of the calmest cruises I went on until we tried to get into Nome. You know, that was something, considering it's September and considering what we've been in before. I think for me and for eventics, for scientists, was the fact that some of these communities seeing adults without the young. And that is a concern to me, even
Clams?
the clams.
Yeah.
But we're also seeing it with these amphipods. There's adults, but they're not the young. And if you don't have any young in your population, you're not going to keep it going. And so that is what we're looking at, size structure. And we have to advertise. We have a paper coming out, Science, next week, in which we show this for the amphipod, particularly that in fact there's some relationship over the last 10 years. They may be abundance there, but they're very, that particular, the biomass is there, but the size structure is changing. And I get that part of it. It's not just one species, it's multiple species. And then I think, and so visually, my comments was something I saw, particularly in this north of Bering Strait, was just a lot of adults. I mean, I can't complain. I like adults and all that, but if there's no young, you're not going to keep that population going. And that is my concern.
What's the life cycle of an arthropod?
Oh, well, in Halfapod, they don't let that, it's maybe 5 to 10. These particular Ampeliscus, they brood their young and they crawl out of their pouches right back into the sediments. And if they don't have, if the sediments aren't the right structure and they don't have the ability to build these tubes, and to build those tubes, you have to have silt. And if the currents have changed and they don't have that, so they're kind of a whammy, there. For the clams, they broadcast, so they put their young into the water column. So what that means is that the fact the young might not be maybe just the adults, is that the youngs need to come from further south, and they're not coming necessarily from the south. So there's a lot, that's something I now have to really look at their life cycle part of it. I do have a past PhD student that's looking at size fraction, but our current studies show it's not just amphipods, but it's also bivalves that they're having smaller, which means that there's less quantity, there's less bang for the buck. That's what we used to say when they dive on the eiders. If they don't have as much of a clam meat, they have to spend a lot of energy to get it, and they come back with less. And that's the same thing that if there's some of the feeding in the southern part of the Chukchi.
So, question, could it be that the amphipods, are they healthy enough, are they eating enough to give young, or to have young?
Well, that's
or are they holding off because they may not be?
That's a good question, and that we haven't really looked the full-on life cycle, so that's we have to look at the life stages on it. It seems to me when we look at the Chukchi Basin seems to have, from what you were seeing, some high productivity there, but there's more copepods. So you may have a nice bloom up there, and the guys down below are going, oh, good food, and the ones in the middle say, we're going to take it from you, and those are the copepods. So the data that we're seeing on this cruise was that there are more copepods, and where I would have liked the food to get down to the guys on the bottom.
Right.
And that's coming. It is something we put in a paper out in 2006.
Yeah.
The fact that climate warming could change the system from being a very benthic system, crab population, a lot of walruses and so feeding, to something more pelagic, more like the southern Bering Sea. And
we see a lot of hunger the last few years, whether it's in seals or fish or birds, not fish so much, birds, seabirds, they can't find the right kind of fish, or there's not enough. Overarchingly, there seems to be, from what we've just seen and experienced here, there's less food.
Yeah. And I've always been food supply is the big driver, it's what I like to eat. And I think we're seeing it in community composition.
Our salmon, which would also be eating amphipods and whatnot, they are getting smaller. A lot of our fish.
Okay.
You have
A question and then we
questions and comments.
Oh.
Can you comment a bit on the coccolithophores? I think of them.
Well, before you go off the,
Okay.
Yeah.
was
hang
there
on.
an abrupt change? Was it a good number of males like the crab, that just quit one place?
Well, I know for the amphipods on this paper that's coming out, it's like 10 years ago, there was just a precipitous drop on this, there was of the size structure of these amphipods. And so that's for the Chukchi Basin because we're looking at particularly related to the gray whales. Gray whales are now in the southern Chukchi Sea, but there's no amphipods there of any quantity that they could feed on, which is why the idea that the euphausias would be in there. So if all of a sudden you come off of this cruise with more questions than you actually went in there for. And that's about what I can say. And so I think this one, you want to say, the only thing I would add to coccolithophore thing is
We're gonna take that in a second.
let's
So hang on for the coccolithophores. Seth has to give us what was his most unexpected item, given your experience, and maybe there's
Well,
nothing, or did you have anything?
I guess, that temperature trace that I showed. When we picked up the mooring last fall, we were seeing that, 21 was pretty cold, and we knew that the ice retreat was fairly late. I was pretty surprised that we didn't get that pulse of warm water being mixed down in October or November. It never showed up.
Last year.
Yeah, last year. So So it's, it's the delayed gratification of getting the mooring back. This year, we still haven't seen it. We'll see what happens, but it's, I think of last year's ice maybe as what it could have been like at this site, back in the early 2000s or so. And I would have expected the water temperature to sort of stick around more like this, but so far, then it bounced back in 2022. There was a lot of there was quite a lot of heat in the water column there. So
Heat in the water.
yeah, heat. So 21, and then we saw a fairly good pulse of heat after that. So yeah, there's something there that still needs to be disentangled.
Yeah, interesting.
Yeah, and Jackie mentioned the weather. It was phenomenal for September.
I know your pictures of rather flat calm water. I know
Yeah,
that's sort of like spooky, honestly.
In those graphs that you showed, where you had you said there was all blue because there was no sunlight on the surface, but you said there was sunlight under the surface?
Yeah, so the sun.
How do you explain that? Not doesn't
Chlorophyll.
rhyme with physics.
So there was sunlight at the surface, and it propagates through the water. But the chlorophyll was not the surface; it was
Oh, the
subsurface.
chlorophyll.
Yeah, the chlorophyll was down.
I was gonna say, yeah, you can't have sunlight not here and.
That's
down
right.
below.
Absolutely.
Yeah.
All right. And we'll get to the coccolithophore question, and this is from Doug. Can you comment a bit on the coccolithophores? I think of them as bad news for zooplankton, seabirds, and salmon production, but perhaps that is only if the blooms are massive. Good question, Doug.
Yeah,
Yeah,
I don't know. Yeah, I think you should take that one.
Jackie's
Well,
gonna
I think
default
that
to take
he's
that.
right, they're right about that. I mean, when I saw that coccolithophore, because they're very small type of phytoplankton, they're also calcium carbonate, so a lot of seabirds have to have a certain size food to be able to get at, and it's the die-off in the southern Bering Sea before were a variety related to the coccolithophore. I don't know who studied any of those parameters when we saw this bloom. We weren't studying, we were going through. If anybody has looked at, went out and did.
Well, I think that's why Marty thought it was a little bit odd that he had seen so many kittiwakes around that day. Because yeah, that was a little bit of a contrast. The first time I remember a big coccolith bloom being in the Bering Sea was in 1998
Right.
when that El Nino was passing through, and I don't think of this year as being particularly warm, but here they are.
Right, and I've always associated these coccolithophores with warmer water. So and so is there maybe a pulse that went through, but the ecological component to that, I don't think it didn't get the same type of studies as that was done. Maybe because there haven't been as many die-offs of certain organisms that strand on the beach. I'm not sure. I mean, I don't know of anybody who's went out there and did studies on this year.
Rick Toman, I think, thank you, Jackie.
Yeah.
Rick Toman writes a response, I think, to Seth. And then we've got Doug again. So for you, Seth, Rick says summer 2021 was anomalously high by recent standards. Sea ice extent in the Chukchi. 2022 and this more modern, typical late summer.
Yeah, that
Okay.
sort of conforms.
And then Doug, back to you, Jackie, says 1998 is what I was thinking about too. Maybe that's to you. Attributed to poor salmon production and seabird die-offs.
That's right.
So did you guys see any, or you guys were just moving through it so fast?
I don't think we saw. I didn't hear of dead floating
Dead
birds.
birds.
No, I didn't hear of any, no, we didn't see any of that. That's what I'm saying, but I don't think, we saw this in the satellite, but there was no like rapid response to look. There was no vessel looking, any type of measurements on it.
Oh, interesting. All right. Lost opportunity, but kind of neat, unexpected. All right, anyone else? I have a question for the genome man, if no one else. Anyone
Are there
online?
other species of fish
Oh,
or
your
sea life
question.
that are coming up because of the warming and the blooms and the mixing of different waters, the warm and the cold?
From my perspective as living here and responding to dead and live marine mammal strandings and just as fishing and things like that. Yes, so we had the water's gotten really warm to the south of us, the ocean has, and the ice comes down every year and tries to, make ice. It's what we're supposed to be doing. And it's having trouble with the weather, with this warm water we're getting. It's having trouble, the sea ice is having trouble sticking around. So it doesn't renew this
what was a thermal barrier separating the southern Bering Sea ecosystem and the northern Bering Sea ecosystem. And so when that failed for a couple of years in 2017, 2018, it was kind of amazing. The overnight, because like Seth, really good explanation about how the water is always rolling north. We had huge predatory, commercially viable fish show up in massive numbers, like pollock and Pacific cod, and it's thrown our ecosystem of the northern Bering Sea into a bit of a lot of pressure, because those fish came up. The temperatures were warm, which was hard for our fish. Those large predatory fish, they're bigger than our small fatty forage fish, so they kind of gobbled them up as well. And there's been a little bit of, I wouldn't say a little bit, there's been chaos in the region as animals are trying to figure out year to year, is that barrier renewed by the ice? We can kind of judge. Last year we had cold weather late. Maybe Rick is listening to this. We had cold weather late, so we kind of actually were making ice more in April, which is unusual. And I think that the cold water underneath kind of got a boost. This year, it's going to be very interesting to see, because the sea ice currently is very far north in the Beaufort Sea. Like maybe, I think Rick has it here, but it's like 400 miles north of Barrow, the ice edge. So now we've got to get that ice all the way down, back down here by when to start renewing that thermal barriers, divvying out two completely different ecosystems. Until then, there's just a lot of pressure, more than normal. That helps.
In the pictures he was showing, there was a thermal barrier that you had quite a pronounced thermal barrier. If the blue was cold.
Yeah, and that's a classic Bering Sea water column structure where upper water column 6 to 8 degrees, lower water column minus
Minus
1
2,
to
yeah.
3, and that transition can happen over 1 or 2 meters. It's the upper water column is warmed by the sun and mixed by the wind, and the lower water column is cold from the prior winter and mixed by the tides. And that just keeps them.
If this
really
was
low.
September, that thermal barrier was still there.
Still
Right,
there.
it was.
It still was.
So it was it looked a lot stronger than I've heard before.
Yeah, no, it was. I mean, that was noticeable because we come in July and it's usually minus 1.6 and 7. And when we came here in September and October, it was warm a couple of years ago, 2 years ago.
Yeah.
But this year it was minus 1.6 south. So that barrier was there, and the ice retreated later this year. You know, that's one thing. We couldn't even work in July in the Northeast Chukchi Sea on the ship that we had because we're knocked out by this ice. And so it was more like a normal, but whether it's normal, whether the ice will come back and make it again, but it was more like 10 years ago as far as the temperature on the bottom. But the biology doesn't necessarily respond on the same time scales.
Right. Yeah, and then all of a sudden, since you've had that, we have this great retreat of the ice in the Beaufort and Chukchi Sea. So we're in a very ice-free Chukchi and Beaufort Sea, even though we had that cold spring. So that's interesting to me. Like, what made that happen? But Rick will talk about upcoming great science. There is another question from Doug, and I think he's trying to follow up here. Is the information you collect coordinated with data from other research cruises? I'm thinking that the Northern Bering Sea survey that focuses on salmon and other cruises that focus on the benthic animals, etc. So these data will be shared with NOAA, and you guys can answer that.
This was a largely NOAA-funded cruise.
Yeah.
Yeah.
So the answer is yes, these data will be shared and are being shared with the NOAA people who do the Northern Bering Sea survey, and also the NOAA people who are doing the salmon, the salmon survey. It's like what's that one called? Not race, but they do not the benthos.
The basis.
Anyway,
Come on, figure
yes,
out what
that's
acronym you
exactly.
want.
Yep, there's a lot of acronyms. No, I'm butchering it. Rick says, as of Wednesday, combined Chukchi and Beaufort Sea's sea ice extent is second lowest of record, only slightly above 2012. So we went from your cold return of cold water, the barrier, to now the second lowest sea ice extent, only eclipsed by the absolute minimum year of 2012 up in the Chukchi and Beaufort. So it has a long trail to come home, and we'll see, maybe this wind. is
So,
gonna-
so that's not super surprising to me. I mean,
Okay.
the Bering Sea ice is not tightly linked with what's going on in the high Arctic. It's doing its own thing.
It's doing its own thing. Okay, that is super helpful. Thank you. That's good.
And all these pictures were from you just landed, so
Right.
they were here. So when you say this was a recent change,
The last
you
one.
are the most recent change we've seen, and it's cold. And you're saying the most recent change we see is that, well, that's sea ice and bottom up and down, yeah.
In the, currently this, I was just talking, sea ice has retreated very
Yeah.
quickly and very far in the Chukchi and Beaufort.
Yeah, well, it'll be interesting as it takes a really long time, then the amount of time to form, like South Pacific Island Appalachia can be reduced. And so whether we'll have, if there's not a lot of brine and temperature increase, so next year we'll be telling,
We'll get
we'll
it
come
next
back
year.
up next year.
Yeah, we want
We
to
are
hear it.
set to come back in August and, so,
Stick around.
Yeah.
Now, we had this gentleman in the back who's been waiting ever so patiently for the sonar
Oh,
affecting whales question.
that's for
That
sure.
would be for Sue.
Well, yes, sonars can affect whales.
Are there certain frequencies that you try to keep to a minimum out there because of that in certain areas?
Much of the sonar that are in use are military use sonars, and they will be using frequencies to maximize their
Not
target
to the
strength
comfort of
and things like
whales.
that.
So
Yeah.
that could put
Yeah.
pressure on the whales, maybe even the young.
Yes.
And what's interesting is we have just in this region, right, had two back-to-back military drills from St. Lawrence Island down. The American drill was August 23rd to September 10th, and that went from, I think that was Operation Noble Defender. And that was from St. Lawrence Island to Shemya to Attu in the Aleutians, and then right up the border, followed by September, is that right? September 14th to the 24th, we had the Russian military drill that was missile operations drill. And that was included our waters, included the Gulf of Anadyr from about St. Lawrence Island, the Gulf of Anadyr, and down into the southern Bering Sea, including the US exclusive economic zone. They could come in our waters, that's legal. And so we've had quite a bit of military
Pressure in that front.
noise probably, right? And then the industrial maritime traffic in the strait was pretty impressive on both sides, but in particularly on the Russian.
Yeah. Do you have any radio tracking collars on whales?
They don't put collars on, but sometimes they stick a lancing
Tag.
beacon in there that's kind of can come off, or they can, if the animal dies, or it transmits when they come to the surface. I don't know right offhand if they've done that recently this year in this region at all, but we'll find out, and if so, we'll usually try to get those guys to come in to
And it was the right whales that you guys are concerned about more than other.
They have not.
it's not there. We're concerned about all whales up
tagged.
here, but there was a slide, yes, about the right one.
Yeah, the right whales have not been tagged. Bowhead whales have been tagged, but I don't know if any of those transmitters are still operational.
And then my question is to the genome, you're the genome guy, right?
I'm a second year master's student, so.
Yeah, no, get in there,
Sorry.
you're in, that's good. Is, when you had the genomics of the invertebrates, what's the end game of that? Will you be able to find a clam somewhere and say, this is a clam from this region, or is it?
Yeah, you can answer those questions with genetic data or genetic data. I'm gonna look at biodiversity, so I'll look at it from the morphological, like how traditional taxonomy, how we've described these animals, and then from a genomic perspective too. So that can highlight whether a population of clams south of St. Lawrence Island is the same population as up in the Chukchi, or if they're diverging. So that can be a question that we answer.
Oh my gosh, okay.
It can also just help us say, like, we've always called these groups of animals the same species name, but maybe they're actually two different species. So it can kind of like tease out some of the trickier.
So if you
yeah.
had, if you had a walrus
Mhm.
and he eats a clam, and somebody gets a hold of that clam in the stomach, and then they asked you, could you find out if you have a really good genetic map of all the clams, could you find out
Definitely.
where he was?
You have to do like, you have to build a primer that
Yeah.
blocks the walrus genome, because there's gonna be a ton of walrus DNA in its stomach.
Yeah,
You can take its stomach contents, and if you have that primer, which is like, that's new science. Like for a lot of these animals that has not been developed.
right.
But you could look at their stomach contents and see, and you could do it for smaller, you could see what a clam's been eating too. What kind of phytoplankton
Wow.
species they're eating, yeah.
Oh my gosh. Okay, we'll see you again. Very good. Pretty cool what you're doing. That's amazing. And what if you had a really fresh clam? Would you have to still signal out the walrus? It's super fresh,
Like if
like the
it
shell's
had
just been ripped off.
eaten it,
Yeah,
I don't know like how, yeah, so it would be better because DNA will degrade really quickly.
yeah.
If it's not preserved. So I think it would be better. I don't know if you could tell how fresh a clam is I've never cut
I
open
can.
walrus's
It's
stomach,
good. Yep.
so-
No, it's possible. Yeah, it is possible.
But I think it would be a good idea to have those species blocking primers
Yeah.
because you might not know exactly how old and you don't want to waste
Yeah.
the resources barcoding.
So I hope that your meet professor is gonna watch this YouTube because you're nailing these questions.
She gave the talk.
Oh, there we go. In person, okay. All right.
One other,
Yeah, Lou.
I know you were talking about comparing the Pacific Ocean, Arctic and Antarctic. Do you compare the Pacific with the Atlantic? Do you go across the Arctic and compare species and things that way?
Yes, actually, there are colleagues in Norway who are running similar types of programs
Yeah.
on the Bering Sea. And in Canada, in the Canadian Arctic. So we interface with them at a lot of different meetings.
Yeah, I would just say, cause, yeah, and these big programs, cause I'm actually on the board for our Nansen Legacy program, they're having their finals, doing a lot of these type of science over in Norway. And with that, and the observing system, the DBO I talked about, has gone now Pan-Arctic, so the Atlantic folks, these big programs are building some time series lines out of their studies so they could start doing what we do in the Pacific. The Canadians have been developing that for the Baffin Bay area and also for the Beaufort. And now Eel Polyakov from UAF and others, and the Koreans are doing one in the East Siberian Sea outside the Russian EEZ, because you can't quite go there. But that's actually pulled into Pan-Arctic connectivity. So, and then we have data meetings. So, but these programs that you see us talking about here, we have our counterparts over on the Atlantic side. And we build those connections and share data.
And Doug Molyneux writes in the chat, the genomics profile will be good baseline as climate change shifts biodiversity. You're right on top of the cutting edge. With that, any other questions? We're over our time. Thank you all for hanging in there.
One question
Go for
for
it.
you.
For you,
The
Yes, sir.
last time that the Sikuliaq was here.
Yeah.
We talked about the people that were looking at the fossils on the early land bridge.
Sarah Fowl, yes.
Yeah. And we don't know anything more about them. They obviously left the boat.
Yes. We're gonna, you're right, Lou, we did have a prelim, what they're going to do, they went off and did it. And actually, that's why, hang on, where we're getting the harmful algal bloom readings to 45,000 cells per liter, which is not a good thing. That was an honest bloom of Alexander. And then the ship had to come in, and then we lost the data from the IFCB because the ship went down to the Southern Bering Sea. I think they had their own adventure down there. But
Well, they had not as nice water, I'm sure.
we should get her back to find out what they did find.
Yeah, I would say they were very exciting because we did hear that they actually got cores down to peat. So they got below all the sediments that have been laid down since the Bering Land. And so they're really
They were
excited about.
getting off the ship. We met them in Dutch Harbor.
Yeah.
Right.
Yeah.
I just remember they were all from the prairies.
Yeah.
And the first thing they left here, it was blowing 20 knots out of the west. And they just, for the rest of the trip, they I want to see how they liked. The
Bering
moving,
Sea.
yeah, the moving land.
Yeah.
There.
Yeah,
Them and Dramamine were very good friends.
they looked well when they came off the ship.
Very good shape.
They were
They
in
were
good
in
shape,
good
so they must have had, hopefully had calm weather too down there, but let's get them back. Yep, we will. So on that one, we'll contact her. Thank you for the reminder. With that, the next Straight Science, we do not have Straight Science next week. We're getting back into the groove though, and on the 18th, we have Michael Cameron. He's the NOAA Polar Ecosystems Program. They are doing the research on bearded, ringed, spotted, and ribbon seals. They're going to give us the most recent results of their research. They're going to talk about their 2024 Ice Seal Aerial Survey, which will be based out of Nome. And they're just going to be thrilled to talk about seals to no end. So with that, that will be on a Wednesday, the 18th. Thursday's a Port Commission meeting, so this will be on Wednesday, the 18th, and that'll be the next one. And after that, we will have Rick Toman, and we'll have a lot of advertisement on that as well. So we've got a good lineup for October and into November.
So I'll be Zoom from here on in.
No, Rick Toman will be in person here. So be ready to come back in or be online, whichever way you would like to hear it. But we're pretty excited about his in-person visit to Nome and his
October 26th, he says.
October 26th. Thank you, Rick. And it'll be in person. So and it'll be online as well. So come in and visit. So that'll be nice.
Sikuliaq is online.
Oh. All right. Hi, Savannah, Dave, Lewis, and Dana. And hello, Sikuliaq. Yeah, you guys got a shout out earlier too. Good. All right. With that, thank you all so much for coming. Sikuliaq, stay on if you want to talk to any of these characters, and we'll say goodnight to everybody here and online. Thank you so much, everybody. Have a good night.